Okay, hello yet again. Intro to Psych students. We're going to do chapter 5 now. Okay, here we are. So chapter 5, sensation and perception. Here's gets back to that old mind, brain, body problem. Sensation is our sense organs' detection and response to external stimulus, energy, and the transmission of those responses to the brain. So eyes, ears, nose, tongue, skin, etc. And perception is the brain's processing, resulting in internal representations. So this is back to Mary, the color scientist, right? So they're not like a camera or another analogy. They very much aren't. Cameras and computers take a picture of whatever it is, just whatever sensory systems they have, so to speak, and then it's usually pretty direct. Ours don't. We process it very early on. Bottom up is what we expect, okay? And top down, higher levels of processing to lower levels. So the prefrontal cortex, choosing to ignore the pain. Now we have certain, again, schemas, right? The cat, this is a figure 5.2 in your textbook. So even though it's not letting me use the finger again. Why is that? I don't know why. Sometimes it does, sometimes it doesn't. So even though those letters are the same, I'm going to, those letters are the same. You see one as an H and the other as an A, given the context of the cat. I'm the cat. So sensory coding here, just some terms. You can get this from your textbook just as much as I can read them off to you here. Environment information to neuronal information. Things get changed over to sodium across axons. Transduction is that process by which sensory stimuli are converted to signals in the brain. And then sensory receptors transduce these physical chemical informations to neuronal impulses. Qualitative and quantitative. And so qualitative is the sense to which they affect you, and quantitative is how loud, how bright, that sort of idea. And here's a picture of green light, gets in the receptors of your eye, goes to the brain, and you know, oh, time to go. Unless you're living Helena and going across to some of the intersections, you want to give it at least 2 or 3 more seconds because people will run the red light. Here's some of the primary visual cortices again. This is your figure 5.3 in your textbook. So here's the taste, smell. I'm going to get back to here. I'm just going to use given that they won't let me do it with my hands, I'm going to just do it with the laser here. So it's vision here, touch, hearing, smell, taste. All these are those are the primary sensory cortices. The where and the what stream, kind of an interesting thing. So it goes from the visual here up to the parietal. Where, remember, the parietal lobe does space, right? It has, this is sense in your body. It does space. And so the where is where an object is, and the what is what the object is. So I can tell you that I have a lamp. That's a lamp there. So the where stream and the what stream came together to say I have a lamp there. You can actually have brain lesions where, so to speak, where this pathway is damaged. So you can say what it is, but you can't really say where it is. And conversely, you can have damage to the what stream, and you can say it's over there, but I can't tell you what it is. So that's pretty interesting, huh? And here we are. Touch is actually more than just pressure. There's haptic, the haptic sense, so it's more than just touch. But it's often just called touch, but it's also pressure, heat and cold. Basically, you can sharp pain, dull pain, that sort of idea. And here's different coding. Okay, neuronal frequency and coding. So psychophysics. Ernest Weber, Gustav Fechner. The idea is to measure the psychological responses to physical stimuli. I say it's a very ambitious idea. Again, it's one of these trying to combine, mind and body. And so the idea was that you could have a response, and you would measure the thresholds. Let's get to that page, and then somehow kind of close that gap, as it were. So sensory thresholds. There's the absolute threshold where about 50% of the time you'll say you saw it or heard it or whatever. So that's the absolute threshold here. Just noticeable difference is you see something changed slightly. A good example of that I always use is seasons, changing in seasons. You'll probably have a time where you go, it's darker now, or it's lighter now. There'll be like a day that you notice that it's different. That's a just noticeable difference, the JND. Weber's law, JND between two stimuli is based on the proportion of original stimulus. Easy example for that. So you're lifting weights. If you have like a 1 pound weight and you add another pound, you're doubling it. So wow, I can notice that. If you have a 50 pound weight and you put another pound on it, that goes from 50 to 51, probably won't notice that as much. That's the so-called Weber's law. So signal detection theory. This is let's see, that's on page 168. And this is figure 5.8 from your textbook. I want you to think about this for a second. So they have hits and misses. So if the response is actually, if the stimulus is actually there on and the response says they saw it, that's a hit. If the stimuli is not there and they say they didn't see it, we'll just keep with vision. That's a correct rejection. So true negative, true positive, false negative, false positive. Now, if you're a person who tends to be a yay sayer, that you say yes a lot, you'll have more hits. Okay, you'll have more true hits, but less. But look at this is one I really want to point out. You'll have more false positives, false alarms. So you say you saw it, even though it actually wasn't there. So think about that for a second. You're perceiving it. You say you saw a stimuli, but it actually was not there. And the reason you think you saw it is that your personality structure makes you want to say, yes, I did. Whereas if you're a naysayer, you'll say that you didn't see it, 55% of the time, over half of the time, when it was actually there. So, I want you to really kind of think about that for a second, because what your response is, what you say your perception is, depending on your personality structure, yay-sayer or naysayer in this case, will change what you perceive of the world, whether or not it's out there. Yeah, so again, talk about top-down processing and schemas. That's really wild. So even though the yay-sayers got 59% of the time correct rejections and 89% correct hits, it was just the opposite in naysayers. So that should freak you out slightly. I hope that freaks you out slightly. And this is the radiologist, trying to make sure that they making the right call. Probably they're going to overcall it just because they don't want to get in trouble. This apparently is some kind of contrast material in the bladder and the stomach, so they're looking for various lesions. Sensory adaptation. This is figure 5.9 in your textbook. I think this is Heathrow Airport, in London. So these are houses that this is British Airways airline, so it's pretty loud. Landing in the airport, which is, I don't know if it's landing or taking off, actually. I never thought of that story. I think it's landing, but it doesn't matter. It's very loud, and they don't notice it anymore. Read synesthesia, last chapter. So does ESP exist? So I probably should move synesthesia up to here. But again, if you see it, you saw it. It's one of the things that they changed. So they talk about BEM study. There's Robert Darrell BEM, kind of Cinder, and a soundproof focused on randomly generated image. He also had one where they had erotic pictures, and they used statistical analysis, and sure enough, people saw them. The arousal pictures more often than chance, but it's probably not true. So think like a psychologist. ESP probably does extrasensory perception, may not actually be true. Might just be statistics. They're kind of screwing things up. Yeah, currently weak or non-existent. Susan Blackmore, who you'll see, see a video of hers, TED Talk of hers later in the semester, studied this for a while, and she was pretty interesting hearing her talk on how she thinks about ESP, because she had a kind of an out-of-body experience, thought it was a really amazing thing, so studied all this paranormal stuff and never got any positive results. And so she felt it was all rubbish, being from the UK. And here's the senders and receivers. So this is a great site. Let's see, I think they talk about it here. Yeah, Luis Alvarez found himself thinking of a long-lost friend from college years. A few minutes later, he came across a friend's obituary in a newspaper. So if you think about a friend and people die, the chances are at some point somebody will have those line up just randomly. Okay, but just by chance this will line up. But it'll seem like, oh my God, this seemed like connected, but probably just a random occurrence. So vision. We're very visual people. We have most visual perception takes place within the brain, not the retina. That makes a lot of sense because perception happens in the brain. We do have a blind spot. So I can do it because if you hold your thumb out about arm's length and slowly move it to the side, it'll disappear right there. Yep, there's my blind spot. Now you don't ever notice it because it's, you fill in the blanks, brains fill in the blanks. But if you have a chance to do that, you got to keep your eye focused on something in front of you. Move to the side, right there. They're good. Kind of cool. It's just right there, and you don't notice it. And there it is. This is a figure 5.13. So you look at the rabbit. Yeah, it's easier to do with your thumb. Here's some anatomy. So your textbook calls it the middle layer. No, wrong. There's the bipolar, horizontal, and endocrine cells. So I kept those ones in there. So cones, rods, and cones. Cones have color, like cone and color. And rods are black and white, sort of density. Horizontal cells and bipolar cells start processing visual material in the retina. That's why they're so important to have in there. Okay, so that's why they're so important. Endocrine as well. So light information is processed here, not the middle layer here with all these different cells. And here's the optic nerve. The blind spot's right here, right where the optic nerve forms and goes up into the brain. This is pretty AMP, anatomy and physiology. I'll kind of warn you. So the right visual field goes to the left primary cortex, left goes to the right. And you see this guy here. What and where pathways again? I guess we already covered that last chapter. And why do we see color? There's three types of cones. That's for blue, green, and red. Okay, there's three types of cones. Remember, cones are color. And this for people who have color blindness. This is where they have that. Some will have red-blue color blindness or blue-red color blindness. This is where that happens in the trichromatic. The opponent process is ganglion cells. They have respond to specific cones and they inhibit each other. So we're going to do something here. This is for color blindness. If you have color blindness, you'll see a number here and not here. And it's usually males that are colorblind because it's carried on the X chromosome, by the way. It probably has some advantage in hunting, has an evolutionary advantage, because people who are colorblind notice changes better, like slight changes in hue. And you'll notice this when something moves slightly better. So the idea from an evolutionary psychology standpoint is that colorblindness persisted because it helped men hunt. So I'm going to do something here. I want you to stare at that white dot. I'm moving my thing out of the way. Go ahead and stare at the white dot. I will actually time this. Keep staring at it. Okay. Keep staring. I'm going to do this for 30 seconds. Keep staring at the white dot. Your eyes will probably drift away, but bring it back to the white dot. A bit longer. Keep staring. Keep staring. A bit longer. Almost there. And boom. Ah, did you see the flag? That was kind of cool, huh? Let's see. Let's stop that. Do it again. Boom, there it is again. Again. There it is again. So that's the opponent process. So the green goes to red, orange goes to blue, black goes to white. Is it bugging you yet? Usually by this point, students are saying stop doing that, but I'm not going to. Some just common things here. Hue, saturation, and blindness. It's a color wheel, you're probably familiar with that. Here's a couple of different ideas. Color subtraction, additive color mixing. Here's figure 5.17 in your textbook. Whereas the insects can see ultraviolet. Humans view the same flowers in visible light, do not see the nectar. Insects see. So nectar is brighter because it's more. And here, this is the TED Talk that you're going to see this week. Really shows this. It shows that this looks like they're different colors, but they're not. Okay, they're not. The color looks brighter than that one. It's not. This is context. This is the top-down processing that happens within the visual cortex. So again, that's kind of an interesting thing. It shows that we have a common hallucination that we agree upon, and therefore we call that reality. Blind sight. Let's say some of the stuff, again, some of the stuff they take out, I kept it in. This is actually Susan Blackmore's drawings, matter of fact. What happens is you have a lesion in the visual cortex. Remember the primary cortex back here? I'll show you that again. Yep, there we are. So primary visual cortex. You get a damage to that one way. Yep, that happened again. Okay. For whatever reason, trauma or a stroke maybe. So you can't see, you can't have a perception. Light does get to the superior colliculus. Okay, the lateral geniculate nucleus of the thalamus is where most of it goes, but some branch of the optic nerve goes down to the superior colliculus. That then goes up to the parietal lobe. Okay? So this is probably due when something catches you at the edge of your eye, something happens and you kind of turn toward it. It's probably that pathway, right? It's that pathway that makes you, oh, what's that over there? So even though they don't have no consciousness of it, they will say it correctly. They'll guess, and they will guess correctly a lot. So I wish it would let me draw again. I really don't know why it does that. Sometimes my computer just doesn't let me. So say you have an arrow pointing that way, and the stimulus points up here. This is a common psychology thing to do. So if an arrow is pointing at where the stimulus is going to appear, you see it quicker because you're already looking up there. And let me get my eraser. Yeah. Back to my pen. And if the arrow is pointing the other way, it takes you longer to notice it because you're looking down there. They did. I saw some people who actually gave a talk on this. They did research on this with blind sight people. So they couldn't see the arrow or the stimulus. They were just clicking on the computer when it came on or not. And even though they couldn't see either of those things, when the arrow was pointing at the stimulus, they did it quicker. And pointing away from the stimulus, they did it slower. So think about that again. We're back to philosophical zombies, right? And they have no conscious experience of that, but yet they're able to interact with the environment. This is a nice little argument that philosopher zombies do exist, and they're among us now. So, object perception requires construction. Gestalt. Let's do the Gestalt. Excuse me. Shape, form, organized wholes, what Gestalt means. This is now processing of visual information. Figure ground, reversible figure illusion, etc. So let's go to some good pictures. This is a classic one. Again, I keep it in here because I like it. So is it two people looking at each other or one person looking at you from behind a candlestick? Now, you probably see both, right? You can't see them both at the same time. It goes back and forth, looking at you, looking at each other, looking at you, looking at each other, looking at you, looking at each other. It probably does that as I say that. They're looking at you. Oh, now they're looking at each other. Now there's a horse with an apple on its nose. That stupid horse. And here's some other ones, just showing some of these principles. This is a figure 5.19, page 179. And here's some more. These are all these Gestalt principles. Okay? So proximity, similarity, good continuity, closure. So even though there's nothing there, let's see, can I draw anything again still? Okay. Even though there's nothing here, it does look like there is. You fill in the blanks. Yeah, you fill in the blanks. It's top down. We can make this look like it looks like a rectangle behind the little column there, right? But if you think of this as maybe solar panels on a space station, okay, you lose that effect. So again, that's top-down processing. Now it looks like a space station or satellite of some sort. Well, that's so you can, again, it's top-down, what you bring to it. Here's a cool one. You can see the blue connecting in there. And you can see the triangle. Let's see, where's my one problem is I don't really see where it is. It's so small. These you can change, again, changing what they look like. Stop back here. I'll do this slowly here. Again, if I could use my finger, which I don't know why not. These are now Pac-Men attacking each other, right? Very mean Pac-Men. By having them be mean Pac-Men, you can almost make the triangle go away. So what you expect to see changes what you actually do see. I hope that's a little bit weird for y'all. I have that one again. There's the from the first thing. Face perception. Fusiform facial gyrus. What do we have on this? He says this is a Thatcher illusion they have. We got the face is so important that certain regions of the brain dedicated slowly to detecting them. Fusiform gyrus, right hemisphere. It's very important. And this is showing that activity. And this is quite a little bit different than the one from your book, but more or less the same thing. You see a face with the eye and the nose, etc. There's not actually a face there. Just look like someone who's really crying. And this is the figure. This is the actual Thatcher illusion. So, oops, come back. So even though, you can turn the mouth, that's upside down mouth. You have to kind of take your head upside down to see it. But again, you put it together to make it look like a face, even though it's really kind of backwards, upside down. And this is the thing for Margaret Thatcher. And if you know who Margaret Thatcher is, I'll be impressed. She was the Prime Minister of the United Kingdom in the early 80s. Mid-80s, she went for the 80s. Okay, some depth perception ones, binocular cues. Here's a fly. Let's get the fly. Here's binocular cues. Green flies going right at you, pretty darn close to you, actually. Yikes! This is convergence. So binocular disparity. You hit different parts of the retina in convergence. And then there's a lot of monocular ones, monocular clues. One eye, interposition, linear perspective, etc. Let's look at some of those. There's interposition. So it looks like the king of clubs is on top of the king of spades. They're not cut out. And there's like a bunch of circles on top of another. Linear. This is why I wish we had, again, this is another reason why I need to go back into the classroom, because I can't really show you this as well. Yeah, those two little creatures are the same height. That's the point. Those are the same height, even though it looks like the one in the back is bigger. And I could measure that for you, but I can't now. Elevation, same sort of idea. The thing that the tree looks like is about the same size. It's actually not. I think I can measure that. Yeah. Shadowing. Again, these are monocular ones. That's all you need to know. Texture gradient. You can see more texture on things that are closer to you. And motion parallax. Things that are closer go faster than things that are farther away. That's what makes it look like the moon is following you. Most people have had that experience when they're driving as a child, and we'll cover this again in development, in the chapter on development, that you think the moon is following you because everything else is zooming by, but the moon seems to be still right there. So it looks like it's following you. That's the motion parallax. And here's the depth cues, position relative to the horizon. Kind of goes up. This is the, let's see, depth perception doesn't show it. Looks like that little tiny girl standing on that poor kid's head. And here's the Ames box and depth perception. It's just a matter of having the box. You have to look at a particular window, right? There's only one little window you have to look through for that. Same idea, Ponzo illusion. The horizontal lines appear to be at different sizes, but they're actually the same. Let's see what I want here. Here's a few out of your textbook. It looks like they're moving, especially if you look in the center of it. And this is a better one. Looks like it's pulsing. And here's the rotating snake solution. This is a figure 5.33. You notice some of the distance snakes are moving. When you look at any one of those things, and clearly it's not moving, but all the ones around them are. Not wild. This is about the time that students will say, please change the slide. And I won't. In various constancies. Yeah. I might do this later. I'll remember to do this later. I'll do it on my phone and then put it onto the YouTube because I can do that. I don't have scissors out here in my office. Although I do have a knife. I'll do that. So different constancy, size constancy. They look like the same size. It's this horizon one again. Same idea here. Size constancy. Looks like they're the same size, but on the retina. And the shape of that one is the same as the shape of this one. Again, shape constancy doesn't. Even though the image changes on your retina, you have top-down processing, and you know that's a door opening, as opposed to very different shapes of things. And this is the one. I can't see this. So I'm going to as soon as I finish this lecture, I'll do I'll get my phone and take a picture of this because I have this on a PowerPoint. You can probably see it here. Look at my little picture in the corner, and I can't see that those are possibly the same. They just totally aren't. But I'll cut them out when I'm done with this lecture and do a little show of that. All right, continuing onward. Audition. Hearing. So amplitude is loudness, frequency is pitch. There's a nice picture of that. This is figure 5.38 in your book. So going up, loud to soft. I'll get back to the laser pointer. And here's the anatomy. Okay, again, this is a AMP, pretty much AMP. Sound goes into your ear, vibrates the eardrum, tympanic membrane, vibrates those three little bones in the middle there, the middle ear, and goes into the cochlea, goes into the auditory nerve. These little hair cells wave back and forth because the sound makes all these waves, right? It makes these waves in this fluid. and the wave in the fluid makes these wave back and forth. That then gets transduced into neuronal impulses, goes into your auditory nerve. It actually goes to both sides of the brain. It's one of the only sensor that goes to both sides of the brain, but it does. And you're able to now hear this gal rocking out on the saxophone. Here's a little bit closer picture of that. That's why I like it. This is also from a previous text. So the middle ear with the hammer, anvil, and stirrup goes to the thalamus and to the primary auditory cortex. And here's even a better picture of those hair cells. Okay, these are these little things here. It's kind of harder to see them, right? But there you do. So the sound wave vibrates the fluid in the cochlea, and those wave back and forth. Do a lot of protein changes here, and sure enough, gets turned into neuronal impulses. Afferent goes at the brain. We can locate sounds. There's a few things here. How to do that? This is now figure 5.4 in your textbook. There's the owl finding the little mouse. In here, which ear it reaches first, even though that's very quickly, if the sound moves relatively fast, the cell phone, you'll get that ear quicker, and you also have a shadow, so therefore you know that the cell phone's over there. Cochlear implants. Now, this is good YouTube if you ever want to take time to watch that. It's pretty cool. But you can put these things onto deaf children. It has to be children to have them hear better, because it basically bypasses the problems with the ear. Now the question is this what's it called? Autism? Where is this? Autism. Where is it? I'm trying to find it here. Yeah. People in the deaf culture thinks this is a way to try to get rid of the deaf culture, because they have their own culture. Autism. And so is this some kind of prejudice that we think you should be able to hear? The deaf culture would argue no, it doesn't, this is part of our culture. So it's an interesting question. If you know people who are deaf, they do quite fine. So we have sometimes we'll get a deaf student and they'll have to have accommodations, for sign language. But then they won't see themselves as disabled, so they won't necessarily fill out the forms, but then they have to get the sign language thing. Pitch, temporal coding, place coding. This shows it here. This is figure 5.42 in your textbook. So high frequencies at the base, low frequencies at the tip, because it flops more, right? Lower frequencies is having more, whereas high frequencies is fast, could be really fast lead frequency up there, and floppy down here for the low frequencies. Let's see. Here's some more gustation. Okay, that's taste. Got the taste receptors and the taste buds going to into the actually medulla and up into the thalamus. Ugh, that thing's gross. Poor kid sucks a lemon. Here's another where the methods in psychology. When you're pregnant, if you have good foods, your kid's going to be more likely to want the good foods too. Become familiar with the taste of foods their mothers consume around the time of their birth. They prefer those familiar tastes. So the idea is, if you're pregnant and you want to have a healthy kid, make sure you have healthy drinks around the time you're giving birth. It makes some sense. Again, this is a study in 2001. The other thing is it's likely that mothers who have good food throughout the pregnancy, and might just be more than the taste of food while pregnant. Olfaction, olfactory epithelium. You just kind of keep going through all the different cells, senses here. Pheromones are basically they're chemicals that go from one person to the next, and you can have you'll smell them and get a certain reaction from them. Olfactory. Let's see. So here's let's get to pictures of those things. So olfactory epithelium goes up into the nose, and when we want to smell something, you sniff it, right? You sniff it. So I want to. It smells like an ink pen because it's a certain smell. You sniff it because the olfactory epithelium is on the top of the nose, the superior portion of the nasal cavity. There it is. Little chemicals go into there, get hooked onto the mucous membrane, and bada boom, bada beam, get changed over to neuronal signaling, and you now get up into your brain and you get to smell the wonderful bread. Haptic sense. So let's see. Yeah. So your textbook does talk about pheromones they play a major role in sexual signaling in many animal species and probably affect humans as well. We'll discuss that in chapter 10. Haptic, temperature, pressure, pain, and proprioception. When you go into, if you ever went to the dentist or something like that, they'll numb your teeth, numb your gums, and they'll ask if you just feel pressure. That's because the lidocaine numbs pain, but does not the pain receptors, but not the pressure receptors. So that's why that is. Temperature, hot and cold. Tactile stimulation of skin is the largest sensory organ. Warm and cold. Those get crossed over if they're intense enough. Cold feels warm, and warm feels cold. And here's a picture from your book. So vibration, pressure, stretching, movement of hair. So I mean, you have different receptors for all this different stuff. And here we have for pressure, pain is free nerve endings. You also have just for skin surfaces. So boom, bada boom, fifth cranial nerve. And wow, they must have won the game. They got the Gatorade dumped all over the coach. So let's see, kinesthetic, location of the body. Let's see, where is that in your textbook? Actually, this is another one that they took out. I always remember as soon as they took out. But this is where you are in space. They have muscle spindles and Golgi tendon organs. This lets your brain know where your hands are and your arms and everything else. This is for dizziness. This is the semicircular canals. They have these little rocks in them, they're called otoliths. This basically means ear rocks. And as you turn your head, those ear rocks rock and roll around, pun intended, rock and roll through the vestibular semicircular canals, and that's where you know where your head is. And pain, so-called gate control theory of pain, that there are pain signals. So yeah, this is a figure 5.49. Actually 5.48 in your book. It can be blocked by larger inhibitory fibers. It's a gate. They have gates for pain. If you itch, if you scratch it, that causes a little bit of pain and releases substance P, which takes away itching. So that's why when you have an itch, you want to scratch. As you see me scratching, you probably have feeling yourself itch too. It's called mirror neurons. So how do you control pain? Lots of different ways. Various medications, non-steroidal anti-inflammatory drugs, Tylenol, aspirin, opiates. But cognitive is better. Think about something else. Focus on something else. Don't, it's just pain. No reason to worry on it. Focus on pain or focus something else and relabel it. So I like to throw in pain scales, the idea of pain scales, because a lot of intro to psych students are going into nursing, and they'll have these pain scales where 0, sometimes they use little faces for kids, but 0 is no pain at all, and 10 is the most incredible pain you can possibly imagine. They're very subjective, obviously. So within a person, if someone had a 7 and then they got better and went to a 3, you can trust that. But between people, you can't really trust it. A 7 to one may be a 2 to another. Okay, so if you're going to medicine and you start using those pain scales, remember that they're very subjective, and within a person, yes, that makes sense. If they were at, or conversely, if they're at a 2 and then went up to an 8, something got hurt. But if one person's 2 and another person's 8, you really can't trust that. So, okay, there we are. That's chapter 5. With the annotations. Takes me a while to get back to the teams. There we are. And I'm going to stop recording. So see you next time.